Let's dive right into this explainer today by asking a truly crucial question about the absolute survival of our global food supply. How exactly do we ensure the survival of essential crops and endangered plants that don't even produce natural seeds? It's this high-stakes botanical puzzle that scientists have literally been trying to crack for decades.
Really think about it. We rely on countless plants for our food and materials. But what happens when a plant is completely seedless, or its natural reproductive cycle just gets totally disrupted? To address this massive challenge of propagating seedless plants and to protect endangered species from the brink of extinction, biotechnology has stepped in with a fascinating solution.
You might picture science fiction when you hear the term artificial seeds, and honestly, they do look a bit alien. But they are absolutely real. Carefully engineered in the laboratory, these bead-like capsules are designed to act exactly like the real thing. They are, without a doubt, a true marvel of modern agriculture.
So to really appreciate this brilliant innovation, we've got to break down the actual biology behind these futuristic plant pods. Let's look at what exactly makes up a synthetic seed from the inside out. The crucial point here is that these rely on what we call somatic embryos. That means they are non-zygotic. They aren't formed by natural fertilization. There is zero pollen meeting an egg in this scenario. Instead, scientists take plant tissues, like shoot buds or cell aggregates, and encase them in an artificial medium. This gel medium acts as a surrogate seed coat and endosperm, supplying all the vital nutrients the plant needs to wake up and grow.
While it all sounds incredibly futuristic, this technology was actually first successfully developed way back in 1982. Scientists Kitto and Janik were the absolute pioneers here, and they achieved this breakthrough using, of all things, simple carrot tissues. Yep, the very same vegetable you dip in hummus actually sparked a revolution in plant biotechnology.
Now, as the technology evolved over the years, it's split into two primary types. On one hand, you have hydrated seeds. These are jelly-like capsules made by dropping an embryo-loaded sodium alginate mixture right into a calcium chloride solution to instantly form a gel bead. On the other hand, you have desiccated seeds. These are coated with a water-soluble resin called polyethylene oxide and then carefully dried into little wafers, which really helps them survive long-term storage.
Okay, so let's move to the next logical question. What's the exact laboratory recipe required to manufacture these synthetic wonders? How do we actually get from a tiny plant clipping to a functional, plantable bead?
Well, the procedure requires a highly controlled step-by-step sequence. First, you establish somatic embryogenesis, which is basically just culturing somatic cells with very specific hormones. Next, you mature the embryos and synchronize them so they are completely uniform. Finally, you mass-produce them, encapsulate them in that protective matrix, and, after a drying phase for some of them, you prep them for actual field planting.
It's an incredible biological journey. You could literally trace the entire process, starting from taking an explant from a basic leaf or stem, growing it in a bubbling liquid suspension culture, dropping it into the calcium chloride bath, all the way to harvesting those beautiful physical calcium alginate beads ready for the soil. Looking at all that complex lab work, you are probably wondering, why go through all this trouble and expense when nature basically makes seeds for free? Well, the benefits are a massive game changer for our food systems.
The advantages are staggering, both logistically and genetically. Logistically, their tiny size offers incredibly inexpensive transport and a really long storage life, completely independent of whatever the weather's doing outside. Genetically, because we're using somatic embryos, you are guaranteeing that every single plant in a large-scale crop is a genetically identical, uniform clone. You can even step in and rescue fragile plant hybrids that might otherwise suffer from total seed sterility.
Plus, think about the capsule itself. That artificial gel matrix isn't just a basic shell, it acts as a highly customized life support system. It's almost like a personalized spacesuit for the plant embryo. Scientists can pack this matrix with incredible additives, essential nutrients, growth regulators, fungicides, beneficial fungi called mycorrhizae, and even antibiotics. It's a totally tailored delivery system designed for exactly what that specific plant needs to thrive.
And we aren't just talking about theoretical lab stuff here. Real-world applications are happening right now. This exact tech is actively being used to save endangered species like the fragile hedgehog cacti. It's enabling the massive commercial propagation of seedless varieties like the grapes you probably buy at the grocery store. It's protecting elite genotypes of super rare orchids and efficiently multiplying drought-tolerant olive trees so we can expand orchard in increasingly arid climates.
Now, beyond just commercial farming and horticulture, this explainer wouldn't be complete without highlighting how synthetic seeds act as a massive insurance policy for global germplasm conservation. To truly wrap our heads around the scale of these conservation efforts, just consider the number 6,000. That is roughly the number of unique potato species accessions currently held by the U.S. Potato Gene Bank in Wisconsin alone. They are actively safeguarding the genetic diversity of one of our most crucial vegetables to protect our future food supply.
By utilizing in vitro conservation methods alongside these artificial encapsulation techniques, these facilities can keep these protected genetic resources viable for years. They use slow-growth techniques, storing the cultures in highly controlled chambers at just 8 to 10 degrees Celsius. By tweaking the mediums, they deliberately slow down the plant's physiological processes. It is literally hitting the pause button on nature. Okay, to be entirely objective today, we've got to examine the sobering hurdles too. Because while the science is absolutely brilliant, there are some pretty steep obstacles preventing the widespread, global commercialization of artificial seeds.
The tough reality is that mass-producing these high-quality embryos is still a very costly affair. It requires highly specialized laboratory skills that you just can't automate easily yet. Plus, synthetic seeds can suffer from mechanical damage during handling, leading to poor germination rates in the field. There's also this persistent risk called somoclonal variation, which basically means the clones sometimes unexpectedly mutate and lose that perfect uniformity we want.
And biologically speaking, it is incredibly difficult to perfectly replicate a natural seed coat. A natural seed is an evolutionary fortress. Our artificial versions are still somewhat incomplete, and unfortunately that leaves a lot of artificial seeds highly susceptible to fatal desiccation, just drying out in the sun, or deadly invasions by hungry environmental microorganisms once they hit the soil.
So that leaves us with a pretty profound final thought to ponder today. As our global climate shifts and natural habitats face unprecedented threats, the demand for resilient, uniform, and easily transportable crops is higher than ever. Will the agricultural and scientific communities be able to overcome these steep production costs and biological hurdles? Can we actually perfect and scale the artificial seed to make it the ultimate foundation of our global food supply? It's a massive challenge for sure, and one that will undoubtedly shape the very future of flora on our planet.